three set of notes is dealing with all those things that Hardy-Weinberg Equilibrium kind of takes as assumptions that don't happen. So what kind of factors are going to affect the way that allelic frequencies work compared to Hardy-Weinberg Equilibrium? Again, it's ideal, perfect situation. We know in real life those don't happen. So there's lots of things that are going to throw that equilibrium off. First of those, mutations. Hardy-Weinberg assumes there are no mutations, that they don't happen. We know that's not true because DNA does it all the time. It happens almost every time you make a copy of the DNA, whether through replication or even through things that are being introduced into the system causing damage to it. So what the things that happen with mutation is it can cause either new alleles or it can switch alleles to some other form of an allele. So it can take one type of allele, type A, and turn it to type B, or it can create type C altogether. There are two main categories by the causes of the types of mutations that occur. First of those are spontaneous mutations. Those are ones that just randomly occur, like a spelling error. You're going along and making a copy of the DNA and replication, and it makes a spelling error. It creates the wrong letter in there. It doesn't get repaired, and that's a spontaneous mutation. Or induced mutations, ones where something from the outside has caused some sort of a change to that DNA. So you're exposed to some chemical that caused that to happen. There's lots of cases and lots of examples of those kind of things occurring in real life. Type of changes that can occur, there's two main types. You can have what's called a forward mutation, where I switch to something totally different. I change it. It goes from A to B, or A to C. I've converted it, I've changed it completely. The letters, if I'm going to letters of DNA, maybe I changed an A to a G, or to a T, or to a C. I've flipped it to another type of nucleotide. The other kind are called reverse, where over time, change occurred, and in the population, it got changed back. So at one point in the population, it became A switched to G, but then later, another mutation turned G back to A, so it went back to the way it originally was in the population. So you've switched the alleles around. That keeps sort of throwing things off. When we talk about evolutionary changes in the population, that's a huge problem because you're trying to calculate how things occurred in the past. When things reverse back and forth all the time, it throws off your calculation of how long ago something occurred. So you have to take that into consideration when they're doing those kind of things. The mutation rate is calculated and is represented by the letter mu. So it's the number of mutations divided by the population size. Number of mutations that occur in a population. That's not the easiest thing in the world to calculate. Sometimes it has to be estimated based upon small sample sizes of the overall population. So it is difficult to measure, especially in recessive traits, because sometimes they're hidden. Mutation rates, are they constant? That's a good question people have to ask all the time. Does it stay the same? Does it stay between different groups of organisms, different populations, different species? The answer to that is there's lots of theories about is it actually consistent? There's not a good answer. Unfortunately, in science, sometimes we don't have good answers because we're always trying to find new evidence to change and look at see how things work. And the short answer is probably not constant. Usually is. For most species within a species, it's usually pretty constant, but it can speed up and slow down based upon factors in the environment. And over time, people have theories about that causing massive changes and showing up new species and things like that over time. The concept between a gradual evolution versus a punctuated equilibrium a lot of times goes back to things like this. So there's no agreed upon answer. So that's the easy short answer to it. So the effects on allelic frequency. How is allelic frequency affected by mutation? Well, it's going to depend on a couple things. Is there a selective advantage or disadvantage to a new mutation? If there is, if it makes it either worse, where all of a sudden that mutation causes that part of the population to go away, that's going to drastically affect it. If it adds one, it becomes extremely more effective, all of a sudden that becomes more of a factor because you have more people surviving to produce more offspring. So generally, they aren't going to have a big deal. The first new mutation's probably not going to have a big issue because they're going to be such small numbers. It's difficult unless you have one of these really extremely good genes or extremely bad genes that causes a problem in the population. So the selected advantage will show the biggest increase. Selected disadvantage will show decreases. Again, if you have one that's going to make you more likely to survive in drought conditions and all of a sudden a drought occurs, that's going to be a huge advantage for you. Your numbers are going to go up in your population. If you have one that causes a fatal disease where all of a sudden you no longer have that part of the population surviving when they're born, they maybe live long enough to pass their gene on and then die, it becomes a big disadvantage. In a smaller population, that is where you can't actually see this. If you have very tiny little populations, then mutations can be a huge deal. So if you have isolated little pockets on like an island, it becomes a big issue because those kind of things become very, very easily swayed by mutations. The same issue occurs with animals in small numbers, like cheetahs and other animals that are highly endangered. That becomes really a problem for them because all of a sudden now a mutation in the population can wipe the whole thing out. There are high mutation rates in some organisms. Bacteria have a fairly high mutation rate. Makes sense. It makes them actually able to adapt a lot easier because they mutate so quickly. So they're able to overcome and adapt to their environment very rapidly because of that. It can also be a disadvantage in some cases. This shows you basically a curve showing mutational rates over generations based on the number of generations that occurred. You see they tend to sort of flatten out the percentage of how often it's occurring. They kind of say it slows down in most species. We use that to calculate what we call a molecular clock. That concept of how long ago did something occur based upon how many changes have occurred since then. The man who came up with this was Moto Kimura, who was a Japanese scientist, mathematician, geneticist, who realized he could mathematically predict on most of these genes how long ago they changed. So you could look at species and say, well, it diverged from this species this long ago based on the number of mutations that have occurred. He uses it from conserved genes, ones that aren't going to change much because if they did, that would kill the organism. So he's going to look at things like histone proteins that wrap the DNA together, because if they can't wrap the DNA together, they fall apart. Or things like mitochondrial genes that are necessary for the mitochondria to work correctly. So they're going to have very slow mutation, very constant mutation, which is going to have very little effect on their overall functionality. He used those to figure out this consistent rate of how long ago something occurred. Human rate is thought to be about 2.2 times 10 to the negative 9th base pairs per year in the population. It's not a lot. It may sound like a lot, but that's a pretty small number. You take the number of zeros in there. So roughly per generation, it's about 277 base pairs out of 6.5 billion per generation that change. Could be big. Maybe that 277 are hugely important ones that affect the biggest genes and most important genes that cause death of the organism. But most of the time it doesn't. We can use this to figure out phylogenies, or family relationship trees. By figuring out how long ago these branches occurred from when they're breaks from each other genetically, we can determine who's related to who, how long ago they diverged. So your stories, things like, well, chimpanzees and humans are about 99% the same, almost 98.7. They use phylogenies to do that based on mutational rates to figure out the number of gene separations over time.